Good morning and good afternoon to you, wherever you are. My name is Anita Ghansah, and I'm a senior research fellow at the Noguchi Memorial Institute for Medical Research of the University of Ghana. Today, I'm going to talk to you about bioinformatics of infectious diseases and the relevance to public health. I'll first of all introduce my institute to you and tell you the steps we are taking to try to incorporate genomics and bioinformatics into infectious disease research and the public health relevance of bioinformatics in the work that we do. And I'll use malaria as a case study at some point. So the Noguchi Memorial Institute for Medical Research is a biomedical research of the University of Ghana. And our core mandate is to conduct research into diseases of public health importance, to train postgraduate students in biomedical sciences and support the public health programs of the Ministry of Health and the Ghana Health Service. We do this specifically through conducting research into communicable and non-communicable diseases. We provide training support in biomedical research for undergraduate and postgraduate students and provide high-end laboratory diagnostics and monitoring and surveillance services as well. So for public health, in order to support the 6 pillars of public health, that is health protection, surveillance, prevention, health assessment, health promotion, and emergency preparedness, we support with diagnosis and then sampling for surveillance. We are interested in knowing the circulating pathogens in our populations and what this means to public health. And then we're also interested in environmental samples so that we know what is circulating in our water bodies, our food, and what this means to public health. So for instance, if there is an outbreak, what we do is to conduct surveillance for cases. And these surveillances could be either active or passive. So we will actively go into the communities and fish out people who have symptoms, and then we test them and then treat them. Or passively, if they have the symptoms, they come to the health post or the clinics and they are tested and treated. So the biological specimen that we take are used for molecular epidemiology studies to support the diagnosis and potentially to look at the relatedness of the cases. And then the field epidemiology bits will suggest the transmission pattern of the infection. Now, there are limitations to these traditional methods. The genotyping that we do in the molecular epidemiology may only tell you the clusters of cases that are present without much information on the order or the direction of the transmission. And the size and grouping of the clusters will depend on the resolution of the genotyping method. And to a large extent, what we are using is just PCR amplification and restriction fragment length polymorphism, which is very low resolution. So we rely heavily on epidemiological investigation to sort of link the cases in order to understand the transmission patterns. And this, for the most part, is very little information that we have. We don't have sufficient epi information to track the transmission. And the diagnosis becomes very time consuming. It's subjective. You get variable performances or results. And so we end up not diagnosing most of the infections. And the phenotyping, obviously, will be very slow. And the surveillance with low resolution genotyping is what relies on very high-ended epidemiological data, which we do not have enough of. So we think that pathogen genomics can help us solve some of these limitations with the current way that we diagnose or we phenotype or we do our surveillance. So we think this is the way forward for us. So comparative genomics analysis by high resolution or high throughput whole genome sequencing offers that high resolution for epidemiological investigations of infectious diseases. And this approach we know has revolutionized outbreak detection, monitoring of transmission dynamics of infectious agents, and antimicrobial resistance across humans, animals, and the environment. So what are some of the ways that we can use pathogen genomics to study public health diseases or outbreaks or pathogens of public health importance? One is understanding the dynamics of epidemics. So what you do is to sequence and assemble the genomes of a pathogen of interest from an epidemic. You build your phylogeny to identify the various clades or subclones and plot the prevalence of subclones across space and time to understand why or how the epidemic is happening. The second way is to discover brand new pathogens. So if there is a brand new pathogen, what we can do is to do metagenomics on samples from a patient with this unknown disease, and then you blast the reads against the database of all known organisms. Look at a set of reads that didn't match to any known organism, and you know that this is the new pathogen. So your sequencing will focus on this unmatching organisms. And after sequencing, you can assemble the genome of this novel pathogen, build your phylogeny, and see how it relates to other members of its genus, and do comparative genomics to identify genes or elements that are present or absent in this new pathogen relative to the other species. And this will give you an idea of exactly how this new pathogen looks like. Now, we are also interested in understanding the drivers and evolution of within hosts and population level evolution of resistance. So at the population level, we tend to look for signatures of selection. It could be a recent occurrence, so selective sweeps within the genome of pathogens that are linked with resistance. And within the host, we look at heterogeneity within an individual with respect to the population. So if there is higher heterogeneity in the individual compared to the population, it may be interesting. This may be associated with either resistance within the clones that the individual is carrying. Now, we can also date the time of viral infection or any other infection. With a sequence data from viruses in a patient, we build a phylogenetic tree where branch lengths correspond to the calendar time and identify the time to the most common ancestor. And this is the time of the infection. And this is normally a coalescent model. And there are different coalescent models that are used to determine the time since the most common ancestor or the time of the infection. The next thing we can do is therapeutic monitoring of drug resistance. So for instance, if somebody is taking antiretroviral, is on antiretroviral treatment, if you sequence the viruses in this patient multiple times and scan the sequences for known mutations that confer resistance to the drug, then you can change the treatment regimen if you find that that individual with time is harboring resistant viruses to the drugs. So moving from green to a resistant red, and then changing the drug so that the new variants are now sensitive to the current drug. Now we can also use pathogen genomics to understand the population structure of pathogens. So here we see a cluster, but different clades within this cluster of MRSA in the UK. These are the two gray clades, and then Germany, represented by Berlin in the white cluster, and then the dark gray representing Portugal. And in the second panel, we are using the tree to understand how the gene flow from Britain through Berlin to spread to other parts of Germany. Yeah. Now we can also use pathogen genomics to understand or to predict resistance. So first of all, you can ask what is resistance. You can look at mutations versus compensatory polymorphisms. And then you can use it to understand the effect of rare variants on resistant phenotyping. You want to see whether a rare variant is now increasing in the population. Is it also conferring resistance now to a drug or not? Now, you must also be interested in what information is in the database and how this database is being maintained. And you must make sure that your sequences, if uploaded onto the database, your sequences should be either new or comparative to what is in a database. And then we are also interested in the quality scores that we will use to identify what is resistant in different modes and levels of gene expression. So quality scores, when we are measuring resistance using gene expression data. Now, diagnosis, we can also sequence pathogens and look for genes that signify diagnosis or markers for diagnosis. Here, the sensitivity in the sequencing directly from clinical samples become very important. Do we have enough quality to give us sensitive, enough diagnostic tools? And for clinical metagenomics, we want to identify which is the pathogen or who is a commensal and which is a contaminant. And then again, we need to build capacity in order to achieve all these. For epidemiology, there are certain questions that you would want to clarify. How can we infer transmission from genomics data alone? How can we infer transmission when it's not just a strain, but a transcriptome that is moving in the population? Do we have enough spatio-temporal coverage of annotated genomes to make proper inferences about the population dynamics of an infection? So yes, you can have genomic data, but the data should be enough to give you actual inferences about the population. Now let's shift to what we do with malaria. These are practical things that we are interested in. So for malaria control, the talk in the malaria community is to eliminate malaria by 2030. I think that's where we are now. And the goal to do this is to come up with discovery efforts at new vaccines, drugs, and to maintain the elimination targets of zero prevalence of malaria parasites in our populations. And to do this, we will need tools for accurate diagnosis, because if transmission is reduced considerably, you will need very sensitive tools to diagnose malaria. And this, we can only use genomic approaches, very sensitive deep sequencing to pick very low infections. And we need effective prevention and treatment regimen. And we need to stop transmission and prevent reintroduction of parasites in areas where we have achieved zero prevalence. during elimination. Now, what do we currently have? We have vector control interventions like indoor residual spraying of homes, and then the use of bird nets. And this is targeted at the mosquito vectors. And we have preventive therapies for pregnant women and children who are living in seasonal transmission areas, and even for populations with seasonal malaria transmission in certain parts of Africa. And we have diagnostic testing with the rapid diagnostic kit, and then microscopy is our gold standard. And we have very good treatment with artemisinin combination therapies. And then we have a very strong malaria surveillance and one vaccine that is at a pilot implementation stage in 3 countries in Africa. These are Ghana, Malawi, and Kenya. But the challenge with malaria control and elimination is the complexity of the malaria parasite. So the parasite is so diverse in its nature because it has to surmount the mosquito host, and then when bitten and released into the body, it has to surmount the liver stage. Even moving from the peripheral blood to the liver stage, it has to surmount the host immune system, and then the red blood cell when the liver stage invades the red blood cell. So it does this with very extensive genetic diversity. It diversifies at various stages, and this makes the whole process very complex in controlling or eliminating the parasite. And as a result, we have resistance to the artemisinin combination therapy. It has emerged and is spreading in the Greater Mekong sub-region around Cambodia, Myanmar, Thailand, Vietnam, Laos, and now in Africa, we have Rwanda and Tanzania experiencing artemisinin resistance on a very small scale. So it is spreading. And then there is insecticide-resistant mosquito vectors, also a major problem. And for our diagnostic kits, there are deletions in the genes that were used to develop this diagnostic kits. And so there are about 10 or 15% of people with these deletions across Africa, and therefore we are not diagnosing malaria properly. But technology is advancing, so we are using nucleic acid amplification techniques for genotyping. We are using panels of molecular barcodes. We started with 24, a panel of 24. Now there are some barcodes that are about 150 for genotyping, microarray technology, whole genome sequencing, whole transcriptome sequencing, and the gene drive. And then we have a huge database of genetic or genomics data. And these resources are very helpful. This is a list of some of the resources available for malaria genomics. Now, specifically, we are doing highly multiplexed targeted sequencing and whole genome sequencing. Our objective is to develop optimal strategies that we can use to look for markers that will help us understand importation, because when we are eliminating malaria, we want to see which markers are coming into the population. And these are also being used to track the effect of various interventions. So if it is a drug or vector control intervention or a vaccine, we are using these spatial temporal monitoring of resistance and parasite population to ascertain the efficacy of these interventions. We are assessing human variation and parasite variation. So we are interested in understanding how antigens are presented
Hello? Hello, can you hear me?
Hello.
Yes, Adekanda.
Was I timed out?
No, sorry. Welcome back, Dr. Ghansah. You can continue, please.
Sorry about that.
Yeah, I guess it's network.
Okay. Yeah. So I was talking about how we were using highly multiplex targeted sequencing to answer specific questions of public health importance. We are interested in modeling and statistical analysis to help us deal with polyclonal infections and large-scale temporal and genetic data sets. We want to preemptively surveil the African malaria parasite for genetic markers of resistance. We don't want a situation where we are overwhelmed with highly resistant parasites. So we are constantly surveilling for resistant parasites. And so we're also looking for new tools for drugs, vaccine, insecticides, and diagnosis using in silico designs that we hope to take through for laboratory validation if we succeed. We're also interested in genetic mapping by linkage or genome-wide association studies to select genomic regions under strong selection. These specifically candidates for drug resistance. And then we are interested in causal variants for highly virulent malaria or severe malaria. We are also interested in mapping studies that can help determine differential expression patterns across the Plasmodium life cycle to enable us to identify new drugs and vaccines.
Anita, sorry, your screen is not shared. Your presentation is not shared full screen.
Sorry about that. Yeah. So these are some of the few studies we have conducted in Ghana to determine the prevalence of previous drugs like chloroquine resistance in Ghana. And it is through these studies that we're able to advise the malaria control program to change the policy from the use of chloroquine to the currently used first-line artemisinin combination therapy. These are molecular epidemiology studies we conducted at our institute. We are still tracking these molecular markers of drug resistance of these previously used drugs like chloroquine, sulfadoxine, pyrimethamine, because we are expecting sensitive parasite to emerge after removal of the drug pressure. And the idea is to advise in the future on either reuse of some of these drugs in combination with other drugs or for prevention of malaria. We are also tracking the frequency of the deletion in the rapid diagnostic kits gene in our population. And we are looking for new genes for diagnosis. And then this is an example of a study that is monitoring the effect of a vector control intervention. That is the spraying, indoor residual spraying in northern Ghana, and its effect on the resistance of the malaria parasite after a period of spraying. And we are interested in the genetic diversity of the vaccine antigen. This red and blue circle I'm showing here is the vaccine strain, the current malaria vaccine, which is made from the circumsporozoid protein. This is the vaccine strain. So it means that all the other strains that we find in these two regions in Ghana will not work with the vaccine. So you see how important understanding the diversity of the malaria parasite is. This means that we either have to develop a new vaccine or incorporate a lot of these strains because the cross immunity that was expected failed on the field. We have also tried to incorporate targeted next generation sequencing in our estimation of drug resistance in our population. And we recognize that this is a very expensive endeavor. So we have come up with innovative tools that will still help us use next generation sequencing by pulling samples from sites. We have succeeded in pulling 100 samples from one site. So we carry to our central lab 2 samples, but a pool of 100 each. We isolate DNA, prepare our libraries, and then we sequence and apply all the bioinformatic tools to determine the prevalence of these resistant markers. And we obtain as high as 99 or 98% concordance when these same samples were sequenced individually. So for population level studies, we think that we can cut costs and still apply next generation sequencing to monitor drug resistance across Ghana. Lately, we are also working with Pine Biotech to understand not just the extent of diversity, but the effect of these variants in our population on the protein structure and its function. And this is one gene, the Plasmodium falciparum apical membrane antigen, which is involved in the invasion of the red blood cell. We want to see how the diverse strains in the population is affecting its interaction with another protein, the PFRON2, that helps with the invasion of the red blood cells. So in summary, what I'm saying is that if we conduct our clinical studies or any field study after diagnosing of an infectious disease, and we have our epidemiology studies and laboratory procedures, we can go ahead and sequence and use bioinformatics in comparative genomics, phylogenetics analysis to help with clinical genotyping as well. Ultimately, to bring about public health action on infection control and prevention and individual treatment. I think I will end here and acknowledge all our collaborators. Thank you very much for your time.
Yeah, thank you very much, Dr. Anita. We have a few questions for you. Can you hear me?
Yes, I can.
Okay. The first question is about okay. Yeah, so the first question is can you share how you analyze data? But I guess you mentioned that.
Yeah, so basically if you sequence your pathogen, you align it to the reference genome and look for any variants within your sequence. And then, so for us, if you're doing drug resistance, after your alignment, you come up with the regions of interest where your resistant markers are, and then you estimate the frequency of your markers of interest. And this is more statistical at this stage, but it is the bioinformatics, the complexity of the bioinformatics involved in attaining good quality sequences and trimming your sequences and doing the alignment and coming up with your variants of interest that there is a gap in doing that, but we know exactly what to do. A few people in my group are able to analyze this data. Yeah. And after getting your frequencies, then you apply any statistics based on the epidemiological question that you or the epi data that you have. You attach it and then you do your statistical analysis. Yeah.
Yeah, thank you very much for that answer. There is another question, and it touches how one can track resistance. I mean, like, can we track resistance applying genomic studies? Yeah, that was the question. Yeah, you answered that, but I guess you want to still add a few things to that.
Yes, I think we have to track resistance. It is very important if we want to be. In the past, when we were using a drug like chloroquine, we were not tracking resistance, and we were just overwhelmed with highly resistant malaria parasites, and we had a lot more deaths in Ghana, I can say that. So with these new drugs, we want to track resistance and control it, or if we find it in a particular hotspot in Ghana, we want to track it and then manage it at that hotspot before it spreads. How can we do that? Surveillance is now a tool for tracking resistance. So in Ghana, we have now 30 sentinel sites by the Ghana Health Service or the Malaria Control Program, where we go there at the moment annually in this drug efficacy studies. You conduct a study, take people who come with malaria, and then you take samples from them. We test for these molecular markers of resistance to these drugs. Yes, at artemisinin, we are still discovering what the markers would be, but any marker that has been found, we are testing. And we also do in vitro testing of resistance in the lab, and then the clinical testing of resistance. And with the pooling of our samples, we think we can do deep sequencing so that we'll pull samples from these 30 sites. And we want to do this monthly, do the deep sequencing so that we'll be able to pick a resistant parasite as soon as it emerges. So when it emerges in a particular region, then we'll focus on that region and do a more extensive study. So it is doable. It's very doable. Yeah.
Yeah, thank you very much for that answer. So like she has said, it is very possible to track resistance. Yeah, I used to remember that as a microbiologist, once before I moved, I became a molecular biologist. We used microbiological ways to also track resistance. And now we have molecular approaches to that. Yeah. So the next question we have here is that good to see your expertise on bioinformatics in genomics. And the question goes like this. It says that of course we get help from software technology to get relevant information and to analyze some things, things about pathogens. But the question is, how much is this? How much specific is this technology? Are they error-free? Have you been able to detect how? The best way to analyze studies that is in this sphere?
Yes, so thank you very much. Like all our traditional methods, microscopy is very subjective. It depends on the microscopist doing it. Microscopy misses a lot of low frequency or low prevalence infections. So with PCR, we were able to account for the errors from microscopy. Now, PCR also has got its own error rating. With high throughput sequencing, you get more depth. It is not error-free, but the depth of information, you're able to pick the most, let's say, just the first few resistant parasite or whatever phenotype you're interested in as it emerges. So you weigh the benefits with the, you want to see whether the benefits outweigh the risk or the disadvantages of the method. So there are advantages, but definitely these molecular techniques are far more sensitive than the standard microscopy or restriction fragment length polymorphism analysis that we were using traditionally. Yeah.
Wow, that's something, and I know after a while we would get to that level of perfection. Before I go to the question relating to ethics of research, I guess I would love to take this next question. It says that how do we obtain statistical information from the public on infectious disease? I believe that this person is trying to ask about the sphere of citizen science and its application to your research.
Did you say statistical information? Can you repeat the question?
Yeah, so the question is, how do you obtain statistical information from the public? I mean, do you go in collaboration with the people? Do you, what public health method do you use to get those your samples and information about resistance and some of these things?
All right. So for every study, whether it is the malaria control program involved or it is a purely research study, you enter the community and you have to, it's called the community entry. You educate the community about the project and you see whether they will buy into the project. You have to make sure that they understand the project that you are conducting, and then you seek their consent. So if it is a study for children, you need parental consent. If the child is over 12 years, you need, in addition to the parental consent, you need the accent of that child. And it is a voluntary thing. Ethically, they are not under any pressure to be part of the study. We tell them, if you don't take part in the study, it doesn't mean you're not going to be treated. So, and there are so many communities that we've worked with, and we have built so much trust. So they work with us in collaboration. We make them part of our study design, or we go to them and ask them what they think we should be doing for them as a community. So they are part of our study, and therefore they're able to collect the epidemiological data from them, and that helps with our statistical analysis. Yeah.
Wow, thanks very much. It's very, very enlightening. I believe that our researchers in the house are taking cues from this. For those that are just joining us, we're on to Dr. Anita Ghansah. Our research includes building capacity in the use of P. falciparum. That's a parasite, and she uses genetic data to describe the extent of the genetic diversity of the parasite. And discovery of new genes and SNPs that has been that is biologically important to research, and she also has been able to adapt bioinformatics advances to further research. The last question I will be asking from you concerning this as we close on this note is the question of ethics. Yeah, you mentioned it, but I would love you to specify in the course of your studies, what major ethical concerns have arisen or or has a likelihood of arising, and how do you go around to solve some of these ethical issues that relates to your research work?
Yeah. So as I mentioned, some of the key concerns that we have with study participants are that sometimes there is misinformation. People think that we are giving them a drug that may affect them in different ways. There was a whole brouhaha about the introduction of the vaccine, the malaria vaccine in Ghana, and we had to do a lot of community engagement, public education for people to understand the importance of the science behind the vaccine and to understand that it was not going to harm them. And there are issues about the risk involved in doing some of these studies. Normally, the kind of samples we draw are very low risk. We tell them this is low risk. But if anybody is affected in the course of the study, that person, there are arrangements for care for that person. And so in terms of the risk to participants, we take care of them. We work with clinicians, and so, but to a large extent, we work on very low risk studies, just either a finger prick sample from an individual does not really hurt them. Yeah. And we make sure that we do not coerce participants into taking, we respect participants' confidentiality. We try to go by the ethical rules for the conduct of research, and we give so much information and our contact so that participants can call you, we the principal investigators or the leaders on the project, for any explanation, and they are free to withdraw from the studies at any point of the study. Yeah. Thank you.
Thank you very much, Dr. Anita. I guess that I will be reaching out to you more in the nearest future. On this note, I would be sharing my screen now. Thank you very much.
Thank you.
If you have other questions, I would love that you write them down, or you reach out to the to your program director or your community engagement manager, because it is going to, I mean, it's something that we would love to answer in the long run.